In the second section of amoeba, I am going to talk briefly about non-pathogenic or commensal amebas. And at the end of lesson, I will say a summary about blastocystis species. 6 species of these amoeba are globally distributed in human. Given their diagnostic importance, the necessity of differentiating between them and their role as health indicators, it is essential for healthcare professionals to know about them. This slide displays images of these 6 species, including Entamoeba coli, Entamoeba hartmanni, Entamoeba gingivalis, Entamoeba mushkovsky, Endolimax nana, and Idamoeba buchli, which will briefly explain in the following slides. The most common commensal species worldwide is Entamoeba coli. Entamoeba coli was first reported in a patient in India. Its prevalence was high in past decades, but has significantly decreased in recent years due to improved hygiene levels in human populations. In this slide, you can see 2 forms of cyst and trophozoite of Entamoeba coli. Size of cysts are vary between 15 to 35 micrometers with 1 to 8 nuclei. Mature cyst that can transfer to human body has 8 nuclei. Very seldom, mature cysts with 16 or 32 nuclei was also reported for Entamoeba coli. Trophozoite with 15 to 50 micrometers in size has short pseudopods with very slowish movement in fresh stool. Trophozoite has single nucleus with excentral karyosome and irregular peripheral chromatin. Transmission of Entamoeba coli occurs through ingestion of mature 8 nuclei cyst forms. In the small intestine, these cysts change into trophozoites. Trophozoites then move to the large intestine where they multiply asexually, transforming into 1 to 8 nucleated cysts. Finally, 8 nucleated cysts exit with a stool and can infect other people. Microscopic diagnosis in medical laboratories is performed using direct and concentration methods with 3 stool samples taken. Since Entamoeba coli is non-pathogenic, treatment is not necessary. If reinfection does not occur, infected individual will clear from infection after some time. Human-to-human transmission occurs in individuals who do not doing personal hygiene. Therefore, personal hygiene, public sanitation, and health education are necessary for prevention. Another commensal amoeba is Entamoeba hartmanni. This protozoan, although have a considerable prevalence worldwide, is usually not seen or reported in laboratories because it is non-pathogenic and its size is smaller than 10 to 12 micrometers. It may also be mistakenly reported as Entamoeba histolytica or Entamoeba dispar. Until 1959, it was reported as a small race of Entamoeba histolytica due to morphological similarities. Its primary distinguishing feature from Entamoeba histolytica and Entamoeba dispar is size. Its size are less than 10 micrometers and its trophozoite 7 to 12 micrometers. Regarding to the shape and number of nuclei in the cyst and type of movement in trophozoite, it is very similar to Entamoeba histolytica and Entamoeba dispar. Its life cycle, similar to the other amoeba, occurs with ingesting 4 nuclei cysts. In the small intestine, cysts change to trophozoites, move to the large intestine, multiply asexually, and change again to 1 to 4 nuclei cysts. Finally, cysts, especially mature cysts in stool, exit to the environment. Diagnosis, treatment, and prevention is similar to the other human intestinal protozoa and amoeba, as you can see and read yourself in the slide. Another amoeba in human is Entamoeba gingivalis. Entamoeba gingivalis is a little different with other intestinal amoeba since it has only trophozoite and is living in its life cycle inside of the gum. It has global distribution. Non-pathogenically is living in the oral cavity, near decayed teeth and pyorrhea gum abscess. Trophozoite of Entamoeba gingivalis is very similar to Entamoeba histolytica and Entamoeba dispar. Its diagnosis requires great attention. As I said, it has only trophozoite form which vary in size from 10 to 35 micrometers. Trophozoite move relatively fast, similar to Entamoeba histolytica and sometimes slower. Transmission of trophozoites is directly human to human by mouth secretion, shared glasses, spoons, toothbrushes, or kissing, and etc. Diagnosis should be done microscopy using samples obtained by dentists from decayed teeth and pyorrhea gum. Staining and culture also recommended if possible. Entamoeba gingivalis is not pathogen and does not require treatment. Prevention should be done through personal hygiene with proper oral and dental hygiene that you can read in the slide. Another amoeba is Entamoeba mushkovsky species, which in reality is a free-living amoeba. Although Entamoeba mushkovsky is normally free-living, but it can also live as a commensal organism in the human intestine. It is quite similar to entamoeba histolytica and entamoeba dispar and can be differentiated by molecular methods. Molecular methods in some countries indicate a relatively high prevalence in human societies. As I said, entamoeba mushkovsky is very similar with entamoeba histolytica. Its cyst has 1 to 4 nuclei with size of about 7 to 20 micrometers. Life cycle of entamoeba mushkovsky is similar with entamoeba dispar by ingesting of mature 4 nuclei cysts. Cysts in small intestine change to trophocyte. Trophocytes in large intestine multiply asexually and finally change to single to 4 nuclei and exit through stool to the environment or can infect humans. Diagnosis, treatment, prevention, and control are similar to other intestinal amoeba, as you can read in the slide. Iodamoeba buchli or Iodamoeba buchli is another intestinal amoeba in humans. This amoeba, also the same as entamoeba coli, is one of the most common commensal amoeba, can live in the intestine of humans worldwide. As you can see, cysts have different shape like egg or ovoid form with a big food vacuole. Cyst has single nucleus with a large karyosome. Size of cysts are vary between 8 to 15 micrometers. Size of trophocytes are 6 to 20 micrometers with a single nucleus and a large karyosome. Trophozoites move slowly by pseudopod. Human can be infected through ingesting of mature single nucleus cysts. In small intestine, cysts change to trophocyte. Trophocytes migrate to the large intestine, multiply asexually, and change again to cyst form. Diagnosis, treatment, prevention, and control are similar with other intestinal protozoa, as you can read again in this slide. The last one, commensal amoeba, is Endolimax nana. This commensal protozoan, despite its small size and often is not detected well by routine laboratory methods, is relatively common worldwide. Cysts of this protozoan are less than 10 micrometers, oval-shaped, and contain 1 to 4 nuclei. Mature cysts being tetranucleated and possessing a relatively large karyosome. Trophocytes measuring 6 to 12 micrometers exhibit short pseudopods for movement and are uninucleated with a large karyosome. Similar to other amebas, life cycle begins when a mature 4-nucleated cyst is ingested. It goes to the small intestine and transforms into a trophozoite, which then moves to the large intestine. There, it multiplies through asexual binary fission, forming a uninucleate to quadrinucleate cysts. The mature cyst then pass from the body in the feces. Diagnosis, treatment, prevention, and control for this organism are also similar to those for other non-pathogenic amebas, as you can read yourself in this slide. Blastocystis species is the last protozoan in this section. Blastocystis is one of the most common intestinal protozoan parasites worldwide. This organism also infects gastrointestinal tract of human. Despite more than 1 billion carriers, the public health significance still remains unknown. Blastocystis has considerable structural diversity. The most common form is characterized by a central body containing 1 to occasionally 5 nuclei, with size of about 6 to 40 micrometers that observe in stool. Additionally, amoeboid, granular, vacuolar, or budding forms are also may seen during its life cycle. In the intestine, it usually has 2 ways in its life cycle. One way transforms into vacuolar form and eventually develops into forms with central body with thin wall. In this form, life cycle repeats within the intestine, known as internal autoinfection. In another cycle, it develops to amoeboid trophozoites and finally develop to thick-walled with central body form, which then pass from body in feces, causing external infection. In most of infected individuals, blastocystis is non-pathogenic and commensal. However, in a few percent, depending on its genotypes, it can become pathogenic and cause symptoms including diarrhea, anorexia, bloating, vomiting, or rash. Diagnosis is microscopy similar to other intestinal protozoa. Treatment for patient presents gastrointestinal symptoms. If necessary, metronidazole is effective. For prevention and control, given its high prevalence and notice to route of transmission, is similar to other intestinal protozoa through personal and public hygiene.